The mic is on? Yes, good. So I'm sorry that my title was rather brief and my abstract was even shorter in the abstract book. I would like, before I reveal the real title, to have a very short comment. So Kevin yesterday gave a very interesting talk, and among a lot of interesting things, he said that we have transferred a socioeconomic model that promotes competition to science. Or more, I'm not sure I'm quoting the exact words, but this was the meaning. But in crystallography, I have a feeling that we're sharing the means of production, the code libraries and so on. We share the results of our labor, like in WWPDB, you heard so much of it, in software like CCP4, but also Phoenix. We collaborate and use transparently the work of others. And if I look at my own background, ARP-Warp and PDB REDO, the two projects that I got involved over the last 20 plus years, more than 50% of what we use in these programs is from others. And also, at the end of the day, the reward system, both in terms of publication and in grants, is based on your peers judging the quality of your work. So actually, it's not that bad. And at least my proposal might be to transfer that system to society and perhaps the financial sector. It could work quite well, or at least quite a bit better than it's working right now. Anyway, back from politics to using homology as a tool in refinement and structure analysis in PDB REDO. So why does this fit in this meeting, which is about model building? Why am I here? I guess because PDB REDO at the end of the day is a refinement system, but it also includes a lot of model rebuilding and a lot of model building, and I think it fits well the theme. Of course, as you might know, my collaborator, Robbie, is the organizer of the meeting, so he could not talk. And perhaps there was some different dark motives for which he invited me. And all the possible dark motives are listed. If you scan this QR code, you can vote for your favorite motive later. So in the meantime, you see the QR code will stay. So the way my talk will be structured is illustrating the problem of bad structures. Why do we need to redo the PDB? Second, I'll talk about how PDB works, and I'll focus to a few things that we use homology for, which is the most recent developments, and then say what you can do with PDB REDO. So something that it's a phrase I like is like, form ever serves function. And it was not said by a structural biologist, but it was said more than 100 years ago. It most commonly attributed in different forms to Louis Henry Sullivan. He's one of the architects behind this fantastic skyline of Chicago mostly. And so you would expect form to follow function, both in biology and in architecture. And this is not always the case because, for example, you know, the form does not follow function. You know, like this balcony, this side chain here is a bit misplaced. You can also see structures that have missing loops. And this is not very handy in any kind of structure. And in other structures, actually, the turns are very short. And these cannot be functional in a protein. In other cases, and I think this was discussed, in coordinating metals, the coordinating water, it doesn't make functional sense in any imaginable way, this construction. And sadly, severe classes in the main chain can also be observed, which can be quite fatal at certain occasions. And another very common problem in protein structures is confusing labeling between what is there and how things should be used. Very common, water molecules very far away from functional sites. And last but not least, chain termination errors, mostly with OXT. Although the OXT example is a bit less dramatic than that one. Okay, so and if you think that these things do not exist in protein structures, actually I would argue that they do. And part of the reason is that don't forget that what is in the PDB was created by different people at different times. And don't forget that PDB is a historical archive. PDB is validating your structures, giving suggestions. They archive what we give them. The models are not methodologically consistent. The refinement programs, the algorithms, the methods, they all changed over the last 50 years. And as a result, the new and improved methods that you've been hearing about in all these CCP4 meetings have never been applied to the older models that still reside in the PDB from the time that there were still like 200 or 300 structures, or even 10,000 structures, or even from last week. And the end point of that is that there are too many problems that can be solved. So this is an example. It's not a very old structure. I'm not going to say what it is exactly, but this is a peptide that is bound in a peptide binding domain, and that's what this paper was about. So if you zoom in into that structure and you switch on the density, you see these. So well, I do sincerely hope that you can all appreciate that this is not exactly what your crystals were suggesting, that these oxygens should be flipped. But now imagine if you're a computational biologist or a molecular biologist that is looking at these structures, so like, oh, this is amazing. You have a carbonyl-carbonyl bad interaction, and this thing is still binding. Like, no, it's not. Just flip the thing there, and you get two hydrogen bonds. So it's problems like that that PDB REDO is trying to solve. And the way that we do things is that we start, of course, with all the models in the PDB that have the associated X-ray data deposited. This is like last year, that was 100%, so which is fantastic. And besides complaining of the missing data, I think the community has been unique on supplying the data and the models. There is quite a bit of data cleanup that's needed, and I'm afraid that Robbie has spent a lot of his PhD time doing that. Then there is the issue of parametrization, of how do you treat B factors, weights between bonds, angles, again, B factors, TLS groups, all these things that you have learned about refinement. And actually then you go and you do the refinement. And that's the point that actually historically, that's how I got interested in PDB REDO, is like at this point we thought like, okay, you did all this refinement, you have the data, everything is working, let's go and do some rebuilding. So this rebuilding is not complete. We don't build a model from scratch. We change side chains, we look at waters, we look at peptide bond orientations, it's quite a few terminal oxygens, loops, you will see quite a lot of these things. And after that, after the rebuilding, in fact, we refine a bit more. And a very big part of that is that we revalidate the model that we had. We see if the validation scores get better or not. And then we make all these models available to the PDB REDO database, which is basically a mirror of whatever is in the PDB, but better. So something that we realized it's 5 years ago, and that was the grant that Robbie had, is that most PDB protein chains, more than 80% of them, have an associated sequence-based homologue in the PDB. If we consider homologue quite strictly on the 70% identity threshold. So low resolution structures can benefit from high resolution, and we were obviously not the first ones to think about it. There are implementations like that in Buster, in Phoenix, in RefMac ProSmart, and so on. The new thing that we did is that we decided to use not to ask the user to assign a specific homologue that can be chosen in subjective ways, but we said like, oh yeah, you know, let's look at all the homologues together and extract information of all the homologues that have better resolution and try to transfer it in the structure at hand. That created actually some interesting networks in the PDB, and because you could see like these are vectors, these are unidirectional vectors, and you see like this is a graph, all these graphs are available interactively, and you can see how information can be transferred between protein families. So that's, for example, a protein family that I spent my postdoc work on, and you see like this structure is receiving information because it's at low resolution or this or this, while all the other structures are contributing information to these low resolution structures. The next thing that we do is that we create these networks, we know what a hydrogen bond is, and we derive hydrogen bond restraints. I will not go on the details of that, all these are published, but the basic idea is that we do get the different distances from the homologues, and we restrict to a target value and a standard deviation that is representative of the distribution of every hydrogen bond in these homologues. To do that, we need at least 5 target values, and we consider unimodal or also multimodal distributions based on k-means clustering. I should say that if we don't have enough homologues, we have a fallback position that we use the 97 different types of hydrogen bonds that I will not enumerate, but they are 97, Bart calculated them, and we use global targets of how certain hydrogen bonds look in the PDB REDO data bank. What this will do at the end, it will mostly improve the definition of secondary structure. For example, you see here, like this is, obviously it's a helix, it looks helical, but you see in the graph, for example, this region was not represented as a helix. Why? Because the hydrogen bond was not correct. If you redefine that with our restraints, this works well. If you look at it in terms of a few different protein families, that's, during my PhD, I was told that's the most important family, alcohol dehydrogenases. You see like this is the mole probability percentile score. It's all over the place. Why? Some structures were low resolution and old, some were more recent and better. If we redefine them, you see that the mole probability percentile clusters to very high values with exception of a few structures. The same goes, for example, for BRCA1 structures, hemoglobin structures. Actually, most this huge distribution ends up in beautiful mole probability percentile, and also for some of other things that we are interested in. So what we did at that point is that we redefined the whole PDB using homology restraints against the whole PDB REDO. And we did not do that iteratively because we saw that one cycle is enough in a small subset, but at some point we did almost 100,000 structures, and which would have taken 60 CPU years, but it was mostly done in 3 days. Everything was completed after a week because of a very few ribosomes and viruses that are in the PDB. And that was a great opportunity to have singularity and Docker images for PDB REDO that you can take and you can run into your lab or into your cluster and so on. Something more new based on that was to build missing loops. And the idea here is that, for example, this is a loop that was in the protein structure. So that's from the PDB. If we put that in PDB REDO, it changes a bit and the loop breaks simply because these 2 amino acids were not consecutive. So somehow the original depositors made them connect, but the sequence was saying they're not consecutive, so PDB REDO broke them up. So what we do is that we look at homologues of this structure, and we can find out that actually there are homologues, and that would fit more or less that density. So what we do then is that we go back and we delete the two terminal residues because they're almost invariably wrong. And then what we do is that we graft all the possible models, we choose the best one, and then we refine it a bit, and you have a beautiful loop with the two glycides missing and the lysine being a bit disordered. If you think that these things do not exist, I'm sure that this is a beautiful water network that could be better interpreted by some chain. And if you refine it, there is no difference density anymore. A few other examples here that perhaps they're not very dramatic. This loop is clearly there. This loop, yes, it's there because other homologs have it, these there. So, you know, there's lots of missing things. There are tens of thousands residues that we added like that. And there's some hilarious examples. My favorite perhaps is this one, that somebody missed a beta strand that was running. So there's a strand here, a strand there, and nothing there. Well, there's a beta strand there, and you can find it out simply by looking at homologs of this structure. Something else that we do again based on homology, but not only, is glycosylation. So for example, there are maps like that. Like here, there's something missing, something. So here, there's an asparagine, waters, and lots of density. That's a sugar. Here, somebody built one sugar, there's a second one, and here, it's not built correctly. These are all things that PDB REDO can now add or correct. And actually, that was a rather difficult project, done a lot through the developments of Paul in Coot that was doing the actual model building and refinement. We were mostly doing the logistics there. And that has been published actually by Bart and Paul recently. The most recent work using homology, and that's the unpublished part that I'm going to show, is LAMA. So the LAMA stands for local annotation of homology masked amino acids, but it's mostly related to an obsession that the PhD student developed after refining a structure of a nanobody that was produced in a llama. He just likes llamas these days, Bart. I even got llama shocks for his PhD, which I'm wearing actually. So anyway. So what does LAMA do? You remember we had all these families that we could relay together. All that information was sorted because we had to do it to derive the homology restraints. So what we can do now is that you can give any PDB code. That's my favorite one, CTN. You remember that? Yes, it's my PhD structure, together with Ivo that's laughing in the front row. And what we do now, in the meantime, there were many more structures of chitinases in the database. So what we do is that, and this you can do for any structure that is in the PDB, this is live. We represent different criteria, like the Ramachandran Z-score for every residue. How good is the Ramachandran plot for this residue? The homology-based Ramachandran Z-score, that's a relative score with a family. And I will not go on enumerating all these, but these are different quality criteria or structural criteria for the protein. And what we're trying to point out is outliers. And I can't remember which talk it was yesterday that somebody mentioned something attributed to Eleanor, if I remember, that if something is an outlier, it's either wrong or interesting. So here, what LAMA can help you do is find in an easy way things that are either wrong or interesting. And how do you do that? So you have like this vertical bar that you can scroll through. And if you scroll through a position that is rather red, that means that something interesting might be happening in that specific residue. So you can click on this residue, and then you get an expanded view that shows all kinds of different information about these structures. So for example, here, this is the backbone torsion C alphas. Sorry, that's the residue type. If it was an aspartate or an alanine, the real space correlation coefficients distribution for this residue between the homologs. the number of hydrogen, of side chain hydrogen bond it creates. So you see, for example, here that in that family, you have 3 types of residues. This is, by the way, aspartate 315, which is the one neighboring to the catalytic residue. So depending on the state of that structure, you can see that it can have none, 1, or 2 hydrogen bonds. The number of contacts with a ligand, in that case, the ligand is a water. Lots of other interesting things that can allow you to see if that's an error or if that's interesting. So you're welcome to use this database. I will not go on showing examples, but we have extracted by looking quite a lot of interesting things, especially for reasonably sized families. And what we're working on, and we hope to have very soon, is that you can upload your own protein structure and look at it compared to all the other homologs from this structural viewpoint. Okay, so going towards the end, something that does not have a lot to do with homology per se, but lots of people talked about fragment screening, where you have the same protein again and again and again, but they are a bit different. So we realize we're in quite unique position to re-refine all the structures, because we have the Singularity and Docker containers, and we can do it in the cloud. We can do that massively parallel. So doing 700 structures should take as long as it would take to do a single structure. And then we can evaluate the maps using tools that are in Qt. And for example, here there is some difference map. You remove the waters, you end up with a nicely fit ligand. There are things that we do certainly different than other programs, not necessarily better. In some cases, we do better, actually. In some cases, not. So it's an additional tool that it's at the prototype level. We've applied it to a couple of projects from our lab, fragment screening, and we hope that we will be able to develop in the coming years. And that's such a result. It's one of my favorite proteins. And these are the different fragments, which are just the way that PDB REDO fit them and refined them. And actually, shockingly, that's a correct fragment there. Okay. Finally, what do you get out of PDB REDO? These are the statistics. They are almost recent, as Robbie is telling me. The bottom line is that if we start with almost 120,000 models that have X-ray data, practically 100% of them can go to PDB REDO. There are very few models that we can still not refine for very weird reasons. 80% of them get a better fit to the X-ray data, as judged by a drop in 3R factor that's more than 3 times the estimated standard deviation of the 3R factor. And 86% get better molecular geometry. I can't remember here the significance criterion, but we were not being, we were being quite strict on ourselves. So, what can you do with PDB REDO? It's not only the database, you can use it for your models. So you can optimize models before publication for work in progress models. You can, there are some new algorithms to visualize the model chains. We like lately what we call the Clivec-like plot. That is a Ramachandran plot with vectors of the residues that moved between the one model and the other. So you see if there are changes within PDB REDO, how the validation criteria changed after PDB REDO. I'm not going to show the pictures. You should run it. It's also it's available as a standalone software package. If you are subscribing to SB Grid, it's one of the packages that's available there. There's also the Docker and Singularity images. Importantly, there's a PDB REDO web server that if you Google for it, you'll find it, and you're more than welcome to submit your models there. It's pretty quick. Actually, that's the website. We have quite a lot of users running quite a lot of jobs. That's how the server looks like, and It's integrating both retrieving things from the data bank and running your own structures. Actually, one of the features is that you can ask, because we cannot be doing the whole PDB with every new version of the program that we have, because it's work in progress. RefMac, which is of course the refinement engine behind PDB REDO, is changing all the time. So if sometimes there are structures that we did not redo for 2 years, it's quite reasonable. We tell you if the structure was done with an old version of the program, you can request to do it and put it on the queue, come back a few hours later, it's redone. So please use the PDB REDO server or anything else. I will not go on, I promise not to go on this graph, but I will have to go on this graph. I mentioned Robbie Yosten several times. He started this project in his PhD together with Gert Vriend. And after that, he joined my lab and we started the rebuilding more than 10 years ago, I'm afraid. And Bart van Boysekum was the PhD student of Robbie that did all the homology work. Martin is the main developer that we go there. And Krista and George are 2 developers. The NTI has been very generous in hosting us and giving very recently a real job to Robbie. The NVO has always supported with career development grants, Robbie and my group. INEX and INEX Discovery have things for development in the fragment screening. CCP4 has very generously adopted Robbie lately, and we're very grateful for that. And also Johnson & Johnson and Westlife have contributed significantly to the project. We're hiring, apply now, not only to work on PDB REDO, but lots of other things. And if you did not get time to vote, the QR code is there. Thank you for attention.
Thanks very much. That was a great talk. So, questions for Anastassis, please.
How do you handle the ligand stuff? You know, do you get a CIF file with your PDB? Because that used to be always true, isn't it?
So when it goes, so it's different levels. So for things that are on the PDB, all the CIF files are there, right? If it's in the PDB, we have the CIF file. If it's something new, you can upload your CIF file. You have to make the dictionary. And if it's fragment screening, we have CIF files we use now the DSI library, the CIF files are there. We will be creating more as we go on.
Oh, yeah. Take it myself.
I think that the graph which you shown that 80% of the structures are better, it's a little bit misleading, because when I came back to my structures, which I've done 5-6 years ago, applying the same RefMac, it just betters with the R factors and everything. I'm not saying that there are no errors in there, but it's just that the software got much better now, so there is an improvement with the R factors and the statistics.
Yes. So PDB REDO is running more than 10 different software modules. By far the most important software module there is RefMac. So if you rerun RefMac on a 10 or 6-year-old model, you might get a lower free R factor only because you run RefMac. If you run it in the context of PDB REDO, that might be even better. Might be or might not be. So certainly this 80% has a lot to do with the developments in RefMac that goes without argument. So thanks for pointing it out.
So Tassos, how do you tell the difference between wrong and interesting?
You look at it. You know, I don't think we don't have any automated fancy computational way other than look at it and see if it's wrong or if it's interesting. No further insight, I'm sorry. It's typically wrong, by the way. Okay.
Again, this is just a comment, but correcting things like the PEP flip makes no difference to the R factor whatsoever in my experience.
Yes, yes, yes. The PEP flips would keep the same 3R, but it's not only so. Actually, I consider much more significant the geometry increase rather than the 3R factor. The 3R factor will get a better scaling. It will get better. Who cares? No, there was it Paul? No, there's something else, Paul. Sorry. No.
The 3R factor will change if you look at the third decimal place.
If you? Oh, the third decimal place,
yeah.
Sure. It might go up, yeah.
So I was wondering if PDB redo can be changed to also take a look at homologs based on secondary structure prediction in cases where you have low sequence identity but very high structural similarity.
It could. I think that it's a very interesting remark. It's not doing it right now. I think that could be a very interesting thing to do for somebody that pushes the red button. So, yes.
I think we'd better move on at that point.
Yes, so you can introduce me for the next talk as well, because I have another 2 minutes for something different,
Okay,
officially, okay. I think. So Capri. So Capri is the assessment of predicted interactions. It's similar to CASP. I'm not involved in it. I'm just a spokesperson because of my involvement in the 3D bioinfo elixir community. So the idea, it's a double-blind experiment based on CASP, which was the homology modeling, but Capri is for predicting protein-protein introductions. So predict the structure of an unpublished complex. So how the experiment is done, the crystallographers are supposed to be contributing data of a new complex structure that you solved. That's why I'm telling you, you're the crystallographers. What happens next is that the predictors have a short time window in which they try to predict the structure of your complex without knowing what it is, but knowing the sequence of the individual components. And everybody can use their methods. And then come the assessors. I think a couple of people here have served as assessors and will be serving as assessors, a few more of them. And they go on and based on established quality criteria, they try to tell how every team, which could be a team of people or a web server, how much better they did. So the whole point of this very short 2 minutes is that Capri needs targets. There's a lot of things that get published and they don't go to Capri. It's not really a big overhead. It's rather easy. There's a website run at the EBI, or you can mail Susanna Hudek with your complex. These structures are handled with absolute confidentiality. There has never been a breach in the history of Capri. You are not going to be scooped because of that. So please contribute your structures to Capri. They really need it, whatever they are, protein homocomplexes, heterocomplexes, protein peptides, protein nucleic acids, and protein sugars. Not ligands is not really a main focus here. So that's the only thing I wanted to mention about Capri. Please, they need your data.
Okay, thanks very much for that. So we move on next to Mark Barden.
So I have to say, sorry, we had 59 responses to the quiz, which is impressive. And most people thought that actually Rob invited me to make sure I signed his contract, which I did sign last year. So thank you for participating to the quiz.